Assalamualaikum warahmatullahi wabarakatuh. My presentation on today is based on a research article titled Determination of Ofloxacin in Urine Using UV-Vis Absorption Spectroelectrochemistry. So, this study focused on detecting the antibiotic ofloxacin in urine using a combined spectroscopic and electrochemical technique.
Assalamualaikum and hi too. So this is our group member. I am AKMAL and just now is ARIF FIKRI. Okay, so now why we analyze ofloxacin. Ofloxacin is commonly used fluoroquinolone antibiotic that treats infection like UTIs and also respiratory illness. And what's important is that around 70 to 80% of ofloxacin is excreted from the body through the urine without being metabolized. That makes urine a useful sample for monitoring the drug in patients. It also helps track environmental contamination since leftover antibiotics in wastewater can cause an antibiotic resistance. So, detecting ofloxacin accurately in urine is useful for both, which is medical and also environmental purpose.
So usually, ofloxacin detection use technique like HPLC or mass spectrometry, which are very accurate but require expensive equipment and sample pretreatment. In this study, the researchers use UV-Vis spectroelectrochemistry, which is a technique that combines spectroscopy and electrochemical analysis. It's faster, cheaper, and doesn't require pretreatment of the urine. They also designed a custom SCE cell which allows easy electrode replacement without changing the optical path, making the experiment more reproducible and practical.
Okay. So now for the spectral data and interpretation, as you can see the data we gave. So in the UV-Vis spectra, ofloxacin shows 3 main absorbance peak at 245, 294 and also 340 nanometer. So uric acid, which is naturally found in urine, also absorbs at 286 nanometer and can interfere with the detection. To handle this, they use tools like cyclic voltograms CVA and derivative voltogram DVA to match spectral changes with the applied potential. This helped them isolate the ofloxacin signal even in the presence interfering compounds.
So at first, they used a univariate model which gave a determination coefficient R² of 0.94, but it wasn't very reliable due to overlapping signal with uric acid. Then they applied a multivariate method called PERFECT, which is parallel factor analysis, which allow a better separation of signal. Using PERFECT, they achieve R² of 0.996, a low detection limit of 13.22 micromolar and recovery rate of 98%. The relative standard deviation was 3.8%, showing good precision and repeatability even with real urine sample. So, multivariate analysis clearly improve the accuracy and robustness of the method.
So now for the advantages and limitations. So this technique has many advantages. It is fast, sensitive, doesn't need sample pre-treatment, and also works well with complex metrics like urine. The custom cell also adds convenience and consistency. However, some limitations still exist. For example, uric acid and other urine components can interfere, so you still need multivariate tools like PERFECT. The method also needs further validation before clinical use. Okay, now for the improvement suggestion. Some suggested improvement include miniaturizing the setup for point of care use, testing on more patient samples, and maybe adding AI or machine learning to automate signal processing. So like slide given, miniaturize the device into a portable version for field or point of care testing. And for the second one, expand testing, which is across more real patients, urine samples for clinical validation. And for the third one is integrate AI or machine learning to automate data interpretation and improve efficiency. And for the last one is combine with other detection techniques such as fluorescence or Raman, or for even better selectivity.
So, in conclusion, this study shows that UV-based spectroelectrochemistry, especially with a well-designed SE cell, is a powerful tool to detect ofloxacin in urine. It combines chemical specificity with spectral and electrochemical data, giving accurate results even in complex sample. By using Perfect, they solve the problem of overlapping signal and make the method much more reliable. This approach could be applied not just in a clinical drug monitoring, but also in environmental analysis in future.
So here are the references I used for this presentation. The first is the main paper by AKMAL itself. The second and third are supporting articles that compare different electrochemical sensors and also explore how pH affects ofloxacin detections. So I guess that's all from us. Thank you.